DL-Amino Acid Labeling Kit enables you to easily label DL-amino acids before HPLC analysis. Let's see how easy it is. The required equipment and reagents are shown here. You don't need to prepare special reagents besides the kit. The kit contains 4 different solutions. First, add 100 microliters of the sample solution including DL-amino acids to a glass vial. Next, add 100 microliters of the start solution and 100 microliters of the labeling agent solution. Mix the contents using a vortex mixer. Incubate the mixture at 50 degrees Celsius for 2 hours. After incubation, add 100 microliters of the delabeling agent solution. Mix the contents using a vortex mixer. Incubate the mixture at 50 degrees Celsius for 15 minutes. After incubation, add 100 microliters of the stop solution. Finally, add 500 microliters of acetonitrile. You have now completed the labeling and can proceed to the HPLC analysis. If you are interested in this product, please check our website for more details.
Nakalai Tesk, the quality for certainty.